This is a truly outstanding conference, just in the diversity of different backgrounds we've got here, and the message that's coming across about digital health technologies. And what I want to do is just share with you kind of the experience we've had at Pfizer in this, and help you orient, and to help you orient kind of where I am in the company. I'm in the R&D organization, and I lead a team of experts both in imaging modalities as well as in digital wearable technology. And so we actually interact across the company from discovery to early clinic. We get heavily involved in the registrational clinical trials, and we're starting to think now about how we utilize digital health in the post-approval space as well. Everything we do is driven by a question that the company has. We're interested in making sure that technologies are really rigorously qualified, that they are validated, and that we have application against our therapeutic pipeline. This allows us to either get early proof of concept or get some more meaningful information about a disease, or have the opportunity to develop novel endpoints. So here's the take home from my talk, really. You know, these are the things we've heard about across the last day and a half, the last 2 days. We need to be able to do this at scale. We need to prioritize and focus on the right ways. We need to focus on, I need to focus on problems that have longevity. So it's no good me going into a project that 6 weeks later falls apart and we exit that area. So we pick carefully where we go. I can't stress how important getting robust data is for algorithm development. We need to listen to the patient. We've heard plenty of that today. Acceptance of new technologies, really need to bring them forward as partners through this. We need to engage with regulators early and often. And then finally, two things: evidence generation, again, very important about how we think about that. And then also thinking about these tools as platforms. So I don't want to just build something for one application. I want to build it so that it has use across the organization in multiple therapeutic areas. So I'm going to share with you today 3 vignettes on this to help illustrate these kind of themes, as you'll see. And these are in nocturnal scratch for atopic dermatitis, some interest we've got in respiratory health, and then I'm going to show you the clinical facility we've built up in Cambridge that allows us to test new devices and new technologies. So atopic dermatitis is a chronic skin condition. It's a really heavy burden on people just with this really serious itching, scratching at night, and that leads to disturbed sleep patterns, which has a knock-on effect to both the patient, and if you're a child, it has a knock-on effect to the patient, to the parents as well. So the question we had there was really, is there a way for us to is there a way for us to quantify nocturnal scratch? So that's one of the behaviors associated with feeling itchy. You're going to scratch, and in a clinical assessment, you'll be asked with a PRO, patient-reported outcome, how itchy do you feel? How's your sleep going? So it's very hard to quantify how you're sleeping, how you're scratching when you're sleeping, right? So this is this unconscious behavior. The question was, can we find a way to quantify this? And the answer is you can, and we've done this very simply with wrist-worn accelerometry. In the studies we've done to qualify the technology, we've recruited patients with atopic dermatitis. We've had them come into a sleep clinic. We've used polysomnography to measure their sleep patterns, and we've also videoed them using these FTIR cameras. So this has given us a really rigorous data set that we can then analyze and then know you're sleeping, and then absolutely identify every motion that we're detecting on that accelerometer and determining that that is a scratch as opposed to just a random hand movement. In the studies we ran, we had people in the clinic for 2 days. We collected all this data. We sent them home for 2 weeks in devices so we could collect data in the home environment. And we had some tools to make sure we knew when they were in bed and asleep. We're able to compare all of that. So that's a really robust data set, but of course we need to compare that to the PROs. We need to ask the patients how they're feeling, and then also compare that to clinical scoring using the ISGA, which is the standard clinical assessment. So with that, we've gone through this and we've qualified this technology not only in adults, but also in pediatrics. So we've managed to get children to do this. As you can see, you don't need to worry about the graphs, but that's showing there's good alignment between the scratch-pi algorithm that we've developed, or the sleep-pi algorithm. In this case, the scratch-pi and its correlation against video annotation. Another key piece here is that in every study we run, we ask the patients for feedback about wearing the devices. If you have a dermatological condition, can you wear a wrist-worn device? Turns out we can. The majority of people said they didn't notice that they were wearing this at all. And then finally, we've taken the data to the FDA and the EMA. They've put plenty of roadmaps out about how to qualify devices and how to bring new endpoints forward. And so we've gone to them, shown them the work we've got. We found this to be very engaging and collaborative, and we've gotten good feedback that we're on the right process. Of course, they say we'd like to see that you can move the endpoint with intervention. So we've started to get to that point now in application. We've used this on some approved therapies that we have, as well as some emerging therapies. In the data I'm showing you here, that we're showing very quickly, we're able to show that you are scratching less and sleeping better while capturing this data undergoing therapy. I'll switch topics now to a second example, and this is where we've been experimenting with voice as a potential digital biomarker. The reason we got into this in 2017 was one of the questions we asked, when we were really thinking about setting up the digital medicine group, we asked across the organization for questions. And the question that came from the vaccines group was, is there a way for us to maintain contact with patients during a vaccines clinical trial? And the reason is, people will go into the trial, they get the placebo or the active therapy, and then we'll never hear from them again. And so your trials have to be very big because you've got to allow for the fact that you have this, what's called loss to follow-up. So the question was, is there a way for us to find a way to monitor people, to detect if they're getting sick early, so that we can then get them to come in and to be swabbed so that we can understand what respiratory illness they have? So we ran a study in a flu season in about 2018 up in Massachusetts. We recruited people who had the flu. We confirmed they had the flu. We had them come in on a regular basis. We had recorded their symptoms. We had them record their voice on a daily basis, and this includes reading a reading paragraph, as well as some phoneme collection. And at the end of that study, we were able to show that there was a clear difference between the data we had at baseline when they were well and when they were sick, when they were sick and when they were well, and therefore we were starting to build an algorithm to be able to detect this. We then started to apply this in 2021 into a trial. As a biomarker, we needed to run a decentralized trial to collect the data we needed in order to get to proof of concept. That was just literally to look to see if we could detect respiratory illness. Of course, there was interest from senior leadership in the company about whether this actually could be something that could detect COVID. So we were asked to expand the study so that we could actually answer that question. And we were given some sensitivity and specificity targets for detecting COVID-19. So the study we ran was fully decentralized. The site PI was Ochsner Health in Louisiana. And the way it worked was this: you brought your own device to the trial. We contacted people through social media. I've got a slide on that next. Once you downloaded the app and enrolled, you went through informed consent, and then you started to collect your symptoms on a daily basis, as well as performing some voice tests on a daily basis on the phone. We also sent you a kit to your house, which contains swabs. And so at baseline, we would swab you and look for COVID-19, RSV, or flu. If you started to show symptoms during the study, we would instruct you to go and swab again, send this in. If you didn't show any symptoms through the study, you would run through 6 weeks, and then we would test you at the end to see if there was any infection. So this was the largest this is the largest decentralized trial we've run, and it is absolutely fully decentralized. The way we did this, we targeted people through social media. We were able to go after areas where we knew that there was a higher incidence of COVID at the time. And we know that we attracted about 2.9 million clicks through social media. This led to about 1.5 million people looking at the Pfizer clinical trial site page, which is where we needed to direct you to download the app. And we had about 54,000 people go to the point of downloading the app. We'd calculated that in order to get our proof of concept target with a 2% infection rate of COVID-19, we need to fully enroll 9,000 people into the trial, and to do this, to give us enough positives to be able to test our hypothesis. But the really good news is we actually managed to do this quite quickly, this study. So people were interested, and we engaged them, and we had all the right tools in place to do this. So a lot of learnings. And so the answer is, we can detect changes in voice, and it does track with symptoms. We achieved the targeted sensitivity and specificity to answer the question about whether this could be a diagnostic. This sensitivity and specificity actually is comparable to a rapid antigen test, which is, again, interesting. We aren't taking this further forward at the current time, but we are using this as a biomarker for its original purpose, which was, can you use this as a technique in a vaccine trial to keep in touch with people so that we can detect when you get sick, so we can go and swab you and understand exactly what illness you have. So a success in many ways. The last topic I got for you today is the Fire Lab. So as we set up our group, one of the things we realized we needed to have was access to be able to generate data on a very rapid way and to try to do that internally. So we actually have built in Cambridge a fully clinically compliant lab where we can bring subjects in, participants in from outside of Pfizer and in the local community, and we can equip them with devices and collect data and compare that against ground truth. This does a few things. It helps us understand if the technology works, understand what it takes to get the data off the device, understand how to write the instructions to be able to use this, both from a patient perspective, but also from a technician perspective. So when we put these into our trials and we put these into the sites, we're able to help them work through how to do all of this and make it as smooth and easy as possible. I don't know if you can click on this. This is an animated, this is just an example, a video of the, if you click on the screen, it might, no. Anyway, this should be moving and animating. It basically shows what we've got in the lab. We've got one of our team walking up and down a gate mat. The gate mat has the footprints appear like Harry Potter. We also have a semi-motion capture system in there, so we're able to very accurately animate the video itself, so we can make precise measurements. So we have the ability to have ground truth both from the gate mat, from the video itself, as well as the simi, and then on either side are traces of data that's being collected. He's wearing a gene-active wristwatch on one hand, so we see the X, Y, and Z movement of the gait measurements as he's walking. He also has a pair of emoticon insoles inside the shoes. So again, we're able to have rigorous data we've collected, allowed to compare it to ground truth, allows us to, again, make sure we've got the robust data we need to start to develop algorithms. Here's some data from the first study we ran. We recruited just a healthy, younger and healthy elderly in. This was to be able to test everything we'd built in the system, both from a SOP standpoint, as well as just seeing if people would engage with it. We had people come in, perform a number of tasks in the lab. This shows you just the gait speed that we've measured, where in the wrist-worn gait pi algorithm that we've written, and you see that there's no difference between a younger and an older group. We sent these people home with devices for 2 weeks so they could live with them in a home environment, and we analyzed that data with the same gait pi algorithm. You can see two differences here. Number one, both groups, the older group now is walking slower than the younger group, but both groups are walking slower than they were in the lab. This, of course, is the phenomenon of any clinical trial where we're having someone watch you, and that's the white coat effect. So we see people performing probably above what their normal abilities are. Again, no difference between younger and older, and walking faster. So this tells us that we can measure gait speed in a home environment quite easily, and now we've got a much more really honest measure of their performance in a typical day, and that allows us to measure this over a continuous basis as well. So again, why this matters: we can reduce subject burden because we've maybe avoided a clinical visit. We're getting a better reflection of study because we're looking over a much more continuous basis than random visits that you would have into the clinic for assessment. So again, I think that one's a success. The last bit of this I'm just going to share with you is, of course, how you take this even further. So one of the areas we've been interested in is pediatric measurements. Pediatric outcomes are rare in many ways. There's not a lot of data out there to show what normal development in children look like. It's a gap in the literature, the one that we've tried to address. And so we ran the study for the MAGIC study, which ran and recruited children between the ages of 3 and 17 years old. Of course, the team had to really think about how you do this. It's unlike doing an adult. You can't just tell them what you want to do. You need to make it fun. So they really had to kind of think about joy here and think about ways to gamify what we want them to do so we can collect the data we want in the right way. So they were chasing after things in the lab, and that allowed us to control kind of speed people were doing things. We had them hanging things on hooks in the lab and various stuff like that. So all of this made it a fun environment for them. And again, this, you don't need to worry about the graphs here, but again, it shows us that the data we've got allows us to qualify our gate pi algorithm so that now we're not just in an adult setting, it's taking us down to 3-year-old. And so this is important to us. This is an age group which is, there's a lot of development, and there's a number of diseases that we've been interested in. For example, Duchenne muscular dystrophy, and now we have data showing kind of what a typical development profile looks like that allows us now to be able to compare that to the challenges of Duchenne muscular dystrophy progression. This brings you back to the beginning again, so hopefully I've helped you understand kind of where we think about this. We do believe that sensors are an important, wearables are an important part of biomarker development for the industry. A lot of what we're working on are things that people have been talking about in this environment over this meeting. And just finally, I just need to thank many people, both within Pfizer on my team, as well as our patients, our investigators who we've worked with, and thank you for listening to me this afternoon.